Hello everyone, my name is Taletrectinib. Today I will be speaking about the drug for non-small cell lung cancer, which is known as Ibtrozi, or with the trade name, which is Ibtrozi, and its real name, which is Taletrectinib. I will be talking about the clinical efficacy of ROS1 positive non-small cell lung cancer of this drug. This is for my class Biotech 457, Games Studies in Biotechnology. So the non-small cell lung cancer burden. So a non-small cell lung cancer is a type of cancer that accounts for 85% of all new cases of lung cancer. Lung cancer is a leading cause of global patient mortality, which is over 85% of the new cases that are in, occurred annually in the United States. The diagnosis for this type of cancer often occurs late stage due to non-specific symptoms such as cough, fatigue, and this is a type of cancer that does not have like a standard set of criteria for diagnosis, which means that the patients oftentimes will find out they're in advanced stages of lung cancer by the time of diagnosis happens. And the need for this drug was that Taletrectinib or Ibtrozi, it targets a specific ROS1 fusion genes that drive the tumor growth. So what does it do? So why are kinase inhibitors? So a kinase inhibitor is a small molecule that is designed to block enzymes, which in this case is a kinase that trigger uncontrolled cell division. So the genetic drivers for this is that the ROS1 rearrangements occur with non-small cell lung cancer, and Taletrectinib is designed specifically to target the specific mutation that I'll be discussing later in ROS1 rearrangement. And it also targets NDRK fusions, which are NDRK 1, 2, and 3, that will also further discuss later. They occur when the genes fuse together. So the efficacy goal is to be reducing cancer cell proliferation and improving survival while reducing the toxicity from the patients. So the current ROS1 inhibitors that are currently approved in the market are first generation inhibitors such as cosanib, asanib, and ritonib. They're considered to be a standard of care that is pioneered for ROS1 inhibition, but they face challenges with emerging resistance, such as resistance to certain drugs such as glucosamine. So the chemical identity of taletrectinib, the chemical name of the drug is 3,4,2-R, 2-amino propyl oxyphenyl, and 1,R,1,3,4-phenol ethyl imazonol 1,2-B pyrazine 6-amine. It and is provided in salt form as dacinib atopy, and the preparation occurs through chemical synthesis by using a succinct and liposomal coupling. The formulation of the drug is designed as 200 milligrams oral calcium that are taken one daily, up to 600 milligrams total. And if you look to your right, this is the structure of the drug. So the first scheme for preparation of dacinib is, according to Zhang et al., is that you start off with methyl coupling of the ara bromophenol and N-bog-dienol, which reaction occurs in the presence of BB3 and DIAD. This reaction yields NBOC 1,4 bromophenol 2R bromoamine, which is used for the right side of taletrectinib. So you attach it and it will be considered on the right side. Following the ionoborylation with this binocle diboron using BDCl2 DBBF CH2Cl2 and a KOAC in dioxane at 80 degrees Celsius. This will give you a corresponding boronate, which occurs with the coupling of 1R3,4-phenol with 3 bromo 6 goro amino 1,2-B benzene, which occurs in the presence of Kf in DMSL at 120 degrees Celsius. This will give you a secondary amine, which is then used with Suzuki coupling intermediate using a BgCl2, which is the BBF with CH2Cl2 and K2Cl3 in refluxing dioxane and water to yield embothascininib with and the protection of carbon amides with using hydrogen chloride MeOH cyanoxane yields taletrectinib-free base, which is finally treated with a B acid in NBROH. And the second scheme is used by Asakorius et al. You couple with 1 bromophore fluorobenzene with the amino using TBO, and NMB at 65 degrees Celsius. This will yield you 1,4-bromophenoxy 2R bromoamine, which upon ampertation with Bach 2O in DHF yields and Bach 1,4-bromophenoxy 2R bromoamine. The formation of 6-goroamino 1 to be buried with CSF in presence of PNED3 and Cl. DMSL at 80 degrees Celsius yields 6-fluoroacimo 1 to be byrosine, and this plan will take you to CH aromatization with bromo compound, which you will use BDOAC2, Otol 3B, and K2Cl3 in degomine at 102 degrees Celsius to yield the diol derivative. The coupling of the intermediate with 1R3,4-phenol ethyl using Na3PO4 in the MSL and successful treatment with ACL yields the corresponding carbazine as hydrogen chloride salt. The interpretation of the 4-imazine 1,2-B benzene 3 euphenol derivative using hydrogen chloride in EDOH at 68 degrees Celsius, which then follows the reaction of resultant hydrogen chloride salt of the free amine with NaOH and EDOH HO water at 50 degrees Celsius, which yields taletrectinib-free base, which you will then treat with acetic acid in EDOH and water. So what is the mechanism of action for this drug? So the drug works by as taletrectinib, or when it was in development, DS6051B, it will completely inhibit the DRK binding sites of ROS1 and the RK kinesis, which I mentioned earlier. And if you look on the image here, right, you can see the drug Taletrectinib or DS6051B inhibits the DRKs A, B, and C. By inhibiting these receptors, you would, as a result, allow for ROS1 G2032R mutation, which is reported in quercetin sensitive non-small cell lung cancer patients with tumors that contain CD74 ROS1 fusion gene. So that means like the first line of treatment for patients does not really work well, as their their cancer is considered to be treatment resistant, and this is located in the kinase, and by using this kinase inhibition by interfering with basically this non-small lung cancer by interfering with this NDRK's mutation binding spots, it will compete with the binding of AGB. But because like of this binding, it will allow acid will bind to arginine residence at position 2032 at the kinase domain. This will inhibit the growth of non-small cell lung cancer tumors. So what was the inclusion criteria for the studies? So the inclusion criteria involved that adults have to be older than 18 years old, and they have to have advanced and metastatic ROS1 and non-small cell lung cancer. So they have to have a ROS1 mutation and non-small cell lung cancer at the same time. The treatment for them was that they will be given oral taletrectinib at 600 milligrams once daily on an empty stomach. They also tested on a low-fat meal, which had different results. And the patients could be treated with cortisone or be the tyrosine kinase inhibitor naive cohorts. And this would use a confirmed objective response rate, ORR, and intracranial activity, which is the brain. And then we'll look for in the brain wherever that non-small cell lung cancer became metastatic. The exclusion criteria for the studies included that untreated and symptomatic central nervous systems or CNS metastasis. It also excluded anybody that had a myocardial infarction within 6 months of enrollment. It also required that the test had to occur using ECG to test for baseline QDCF or QG interval prolongation that was greater than 450 milliseconds. It also excluded patients that had active HIV infection, hepatitis B, or hepatitis C infection. It also excluded any patient that suffered from congestive heart failure based on New York class 3 or 4 heart failure. So how was the study flow and identification done? So the trial enrollments happened in phase 1 and phase 2. Phase 2 clinical trials were TRUST 1 and 2 clinical trials. Phase 1 clinical trial was done in the United States. In the phase 2 clinical trial, they pulled the results from TRUST 1 and TRUST 2 data, and they evaluated the efficacy of the drug. For phase 1 study, which occurred in the United States, they had 46 patients that they established the dosing and safety study in. In those 46 patients, they using the phase 1, they were using the 3+3 dose escalation design, testing levels from 50 milligrams to 1200 milligrams. Based on the phase 1 clinical trial, using the data from that, they settled on 600 milligram dose for phase 2 clinical trial. And for phase 2 clinical trial, they used 273 patients across the global centers for phase 2 clinical trial. So what were metastatic tumors? Non-small cell lung cancer in the brain, basically. So patients that are treated with first generation kinase inhibitors progress due to off-target bypass pathway activation. So basically meaning any mutation that will allow the cancer to get around and get in the brain as the brain was friendly place for the cancer to be. And according to BIOT et al., the Senate demonstrated in 17 kinase inhibitor naive patients with measurable baseline brain metastasis. ICOR was 76.5%. That means that the drug was very effective at shrinking tumors inside the brain and allowing the tumor shrinkage to occur at 76.5% is a very good result. So what were the adverse reactions? So the most adverse reactions were diarrhea, nausea, and vomiting as the most common symptoms. There were also some cardiac events, such as 19% experienced QD prolongation and 88% experienced validated ASD and 85% observed ALD. And there was also symptoms of dizziness in dystonia, which occurred in 46% of the patients. So how was the toxicity management and modification done? So the starting dose was at 600 milligrams once daily, but if the reduction needed to occur, it was reduced to 400 milligrams once daily. Second reduction was 200 milligrams once daily. And the management action for the trials was hold or discontinue if grade 3 or 4 toxicity persists. So, in conclusion, the trial showed that the Taletrectinib Ibtrozi is a robust next-generation inhibitor with high efficacy in brain metastasis and cathepsin-resistant mutations. It also showed that the PFS exceeds 49 months in naive patients. And the safety data showed that it is manageable drug with proactive monitoring. And it also has a best-in-class potential as it will be a new standard for ROS1 plus non-small cell lung cancer care. Thank you for listening. Hope you have a good day.